Hello students, today we are going to discuss UV visual spectroscopy. It's it's a molecular spectroscopy that is involved the study of interaction of ultraviolet and visible radiation with the molecules. In my previous video I've discussed how electromagnetic radiations actually interacts with the molecules. Similarly, the UV light and the visible light, they have just the right energy to cause electronic transition of electrons from one filled orbital to the another. So when a molecule absorbs light of appropriate wavelength, the electron is promoted to the higher energy orbital. The molecule is then called to be excited. Now, the wavelength of the light that has been absorbed, which is a characteristic of a particular molecule, this gives information about the energy gap and the functional group related to it. So UV region, it ranges from 190 to 400 nanometer, and visible region is from 400 to 800 nanometer, and the technique can be used for both qualitative and quantitative test. The spectroscopy is based on Beer-Lambert's law. The Beer-Lambert's law says that absorbance is proportional. I'll be discussing instrumentation in my next few slides, in which what we will be doing, we will be having a sample and we are going to pass light through it. Now, there will be some absorbance when the light passes through that sample. So the amount of light absorbed depends upon the concentration of the substance. Means if solution is more concentrated, it will have more absorbance. If it is less concentrated, it will have less absorbance. And it is also based on the path length. So according to Beer-Lambert's law, the absorption depends upon the concentration as well as the path length. So this can be expressed in this form, like A refers to absorbance. This is eta, which is molar extension, concentration, and the optical path length. Now this path length is generally kept constant, so absorbance is actually dependent on concentration. If the absorbance of a series of sample solution of known concentration are measured, you can draw a plot of absorbance versus concentration, and which is known as calibration graph. This figure shows the calibration graph of the absorbance and the concentration. This is actually has to be a linear graph. This is the pictorial representation of the instrumentation part. We have the light source, we have the lens. This lens actually converges lights to this monochromator, which is a prism or it can be a grating. Now this monochromator splits the light into individual wavelengths, and this slit actually allows the appropriate wavelength to pass through it. This appropriate wavelength is then passed through a sample solution through which absorbs, and again, as I discussed before, this absorption would depend on the concentration of the sample. After passing through this sample, we have this transmitted light, which then finally goes to the detector, which gives a digital display of their readings of it can be absorbance or it can be set at transmittance also. This is a diagram of a double beam spectrophotometer. As you can see, here is a light source, and this, with the help of this diffraction grating, the light is focused on the two mirrors, and we have one as a reference beam and one as a sample beam. So reference beam, generally in reference we keep the things in which the sample is made. For example, if it is media, it's a culture, then we can keep media or simply water as a reference, so that the absorption because of this blank is nullified and we have the reading of only the cells of whatever analyte we have in our sample. Light source for UV visible spectroscopy is generally tungsten, or it can be a deuterium lamp. The light is then focused with the help of diffraction grating or the prism into which separates this light into individual components. This is the diagram of deuterium lamp and the tungsten lamp. The sample holder for UV visible spectroscopy is known as cuvette, and can be made up of silica. Then the light finally goes to the detector, which converts the incoming light into current, and then a chart recorder helps in plotting the absorbance against the wavelength. Coming to the applications of UV visible spectroscopy, this helps in determining the concentration of the substance, and this helps the scientists in labeling the rate of a reaction. Like if a reaction is proceeding in forward direction or it is proceeding in reverse direction, this can be determined with the help of UV visible spectroscopy. Similarly, we can study the reaction kinetics. The rate of change of concentration of reactants or the products can be determined, and by plotting this absorbance against the time, one can determine the order with respect to the reactants, and hence the rate equation can be determined. Similarly, in case of biochemical and genetic fields, you can determine, quantify DNA or the protein or the enzymes. In case of dyes, ink and paint industry, UV visible spectroscopy is used for quality control. In environmental and agriculture field, quantification of organic materials, heavy metals in fresh water, this can be carried out because all this depend upon the concentration. And UV visible spectroscopy follows Beer-Lambert's law, which actually relates absorbance to the concentration. That was all about UV visible spectroscopy. Thank you.